Good day everyone. My name is FanechkaEsterhuysen and I would like to talk about one-dimensional convolutional neural networks for the classification of protein sequences. Machine learning refers to the design and application of computer models that learn from existing data to perform prediction-type tasks. Deep learning is a type of machine learning that uses artificial neural networks. While traditional ML algorithms are linear, deep learning algorithms are stacked in a hierarchy of increasing complexity and abstraction. Diverse deep learning algorithms have been used to address a wide range of biological data analysis problems, and one of the most challenging applications involves protein classification, as well as protein structure and enzyme substrate prediction. This information is of significance for the pharmaceutical and food industries, amongst others. The primary protein sequence, which is the sequence of amino acids that make up the protein, resemble text. And therefore, the first machine learning algorithms use natural language processing for protein classification. Natural language processing is the application of computational techniques for the analysis and synthesis of natural language and speech. Spell check in Word and autocomplete on our cell phones are some of the daily applications of NLP. In science, NLP is used for text mining, as you do regularly when using Google Scholar. While text mining would focus on analyzing the letters in a word or words in a sentence, analysis of amino acid sequence of proteins aim to identify sequence motifs that are relevant for protein structure and function. Natural language processing is conducted using deep learning algorithms such as recurrent neural networks. An application that we are all familiar with is text prediction on our smartphones. Here's an example of an RNN. For text prediction, the RNN is trained with thousands of sentences to identify the most probable words to complete a phrase. The predictive ability is achieved by repeatedly looping through word combinations of provided sentences and modeling the most likely outcome for a particular phrase. So it is raining is most likely followed by outside, today, or cats and dogs. Another type of deep learning architecture is convolutional neural networks. They are usually applied for image analysis, for example, face recognition. Here the image is divided into smaller sections with a defined number of pixels that permit matrix generation and feature extraction. As the CNN moves over the image, it starts combining different features, example eyes, nose and mouth, which will eventually permit object classification. And typically several layers of extraction are completed, which not only increases the number of extracted features, but also informs on their special relationships. With regards to protein classification, convolutional neural networks differ from recurrent neural networks in several ways. CNNs capture features without position dependence in pattern-like fashion. RNNs do this sequentially and with position-dependent feature extraction. Convolutional neural networks are more robust due to their innate receptor fields and can be run in parallel, whereas RNN has memory constraints and can only move in amino acid sequence order. The aim of my project is to develop a 1D convolutional neural network for the classification of diverse plant protein families. A one-dimensional CNN applies that the amino acid sequence is provided as a one-hot vector as shown here. The proposed architecture has been adopted from a previous publication by Salkai and Gralmus in 2017. To establish proof of concept, the prototype will be trained to classify membrane proteins using a published Swiss Prot dataset used in a publication by Guo et al. In 2019, and thereafter the CNN will be optimized for classification of other plant protein families. With that, I would like to thank my supervisor, Dr. Liana Hesse, the University of the Western Cape, the NRF, and the CHPC.